sorry, we're trying to do this as a joint presentation. And so getting the kinks worked out of remote control. Okay, so I'm Michelle, and Baoxing is also going to present. He's the other video visible. And we're going to talk today, so to introduce ourselves, we're both postdocs in the Buckler lab at Cornell. Baoxing is primarily interested in comparing genomes to one another in a lot of ways, and that'll come out in everything that we present today. And my focus is on transposable element evolution. And we're going to be presenting work from a recent paper. And as long as this works, there, it advanced, good. Oh no, okay. So we're going to be presenting work from this paper, AnchorWave, which is a software that performs pairwise alignment of genome sequences to one another. And we're going to highlight how it really is functional for genomes that have high sequence diversity, a lot of structural variation, as well as differences in rates of whole genome duplication. So once the slide moves, we're going to talk about how genome alignment is really essential for understanding anything we want to know about a genome. Once we have sequenced a genome, we have a string of base pairs, but we don't really know anything about them until we start comparing these sequences to other individuals, to other species, to other genera. And the prevalence of genome sequence has really just been expanding over the last 2 decades, shown here on the right, as the number of plant genomes that have been sequenced in the last 20 years. And so this represents about 0.2% of all green plant diversity. And recently, the Earth Biogenome Project has kind of put out this moonshot goal that they're going to sequence 100% of all plants in the genome, or all plants on Earth in the next 10 years, which kind of sounds amazing and ridiculous, but it does motivate a lot of the things we're doing for comparing genomes to one another. Because once we have all of those genomes, we need to be able to interpret them and understand them. And aligning genomes is kind of the first step to doing any of that. And so a lot of genome sequencing is initiated in animal genomes. And so that meant that a lot of the bioinformatic development of genome alignment software was done with animal genomes in mind. And plants have caught up, but a lot of these kind of fundamental initial algorithms and approaches were designed thinking about animal genomes. And there are some major differences between the ways that animal and plant genomes are organized and kind of the architecture and evolution of those genomes. And these specifics that I'm going to highlight today are from a paper about 10 years ago. So some of the details have maybe shifted a little bit, but in abstraction, all of these things are pretty true, that of these 4 major differences between animal and plant genomes. So the first being that animal genes are much longer than plant genomes. Here, this estimate of about 40 kb of an animal genome versus 3 kb of a plant genome. And like any of us that are used to looking at kind of plant genes, if you open up the human genome browser, you're just kind of shocked by just how big those introns are. It's just pretty obvious that there's something very different with the way that genes are organized in these taxa. Another thing that's really important distinguishing how genomes are organized is the fact that transposable elements were active at different times in the evolutionary history of these taxa. So transposable elements are these pieces of DNA that can jump from one position to another. And so when they're doing that, they're making repeated copies of themselves at dispersed locations throughout the genome. And in animals, that activity was really old. And so that means that there's been enough evolutionary time for those repeats to diverge from one another and be somewhat different. So we don't expect them to be completely identical. Whereas in plants, all of this TE activity has been pretty recent, such that those dispersed copies are identical and can be pretty confusing for confusing and complicating for alignment. Another major difference is regulatory sequence in animals is usually much longer and more highly conserved. Like if you look at the ensemble tracks of the human genome, you can see kind of promoters and enhancers conserved all the way down to zebrafish and sometimes even further. Whereas in plants, a lot of this regulatory sequence is shorter and less conserved, oftentimes because transposable elements have jumped in and kind of stretched things out. And then a final difference between these genomes that we'd be remiss not to highlight is the fact that plant genomes have a whole lot more polyploidy and whole genome duplication than is found in animal genomes. And when it is seen in animals, it's pretty rare and very old. And so I've just talked through a lot of words on this table, but we can also look at this pretty visually. Here in a paper from Zhao and Schrantz in 2019, looking at the level of synteny in animal genomes on the left, as exemplified by mammals, and plant genomes on the right, here, angiosperms. And so in this paper, they were looking at synteny between these genomes. And as plant biologists, their definition of synteny is a little bit different than what we're used to. Here, they're using just kind of exact gene microcollinearity. So if you have genes in order along a chromosome, if even just a single one is missing in one or both species comparisons, that's considered non-syntenic. But even with kind of this different definition than we're used to, it is pretty clear that animal genomes look very different from plant genomes, in that this heat map of comparing every species to every other species on the left is largely orange, showing a lot this high level of synteny, whereas in plants, this heat map is mostly blue, largely because these genomes have had these whole genome duplications and fractionation. And so the thing is that a genome alignment software that expects something like we see on the left probably isn't going to perform quite as well when we give it a genome that looks much more like something on the right. And so, so largely because of that, these methods don't necessarily work. And now Baoxing is going to take over to talk about kind of why they don't work.
So here I'm showing like the results between like the genome alignment with maize and sorghum. So the maize and sorghum are two like related species divided about 12 million years ago. And we will perform alignment using a pipeline widely used to align human genome and champion genome. And we could find only like 4% of maize genome could be aligned to sorghum, but it also looks like there are a lot of regions could be aligned. But if we look into the base pair, only 4% could be aligned. And also another problem is we here the right part is a very nice plot from Dan Snable about 10 years ago. And he shows there's a whole genome duplication happened with maize genome after division from the common ancestor with sorghum. And for the existing whole genome alignment approach, we could either perform a one-to-one alignment or many-to-many alignment, no matter one-to-one or many-to-many. This is different with the known whole genome duplication, so we could not use existing tools to perform genome alignment in cane synthesis with the well-studied whole genome duplication evolution history. And we also looked further why very tiny proportion of maize genome could be aligned to sorghum genome. So for the existing genome alignment tool in general, we use this kind of seed and excision alignment strategy. So firstly, for the reference genome, we generated this kind of keymarks, and we find this kind of shared keymark between the reference genome and the query genome. And for this kind of we use this kind of keymark as seed, and we do the extension using the dynamic programming algorithm. If there are some repeat sequence in the genome, this kind of maybe we could generate some a false positive seed and generate false positive alignments. And also for the transcription factor binding sites of plants, they are like functionally important elements, and we think that their sequence are also conserved across different species. However, their core motifs are only around 7 base pair on average, so this is much smaller than the chimer size we use to trigger the alignment. So for the chimer size, it's not sensitive enough to perform alignment in this kind of very important regular elements. And for the excitation step, in general, we use this kind of like a dynamic programming algorithm. If you ever heard about this kind of Smith-Waterman algorithm, like when we perform a sequence alignment for 2 sequences, we like put one sequence at the top and another at the left, and we fill out this kind of score matrix by like, for example, if you for like a cell, you get a score from the diagonal direction, which is a match or mismatch, or from top, which is an insertion, or from left, which is a deletion, or also we could also compare with a 0. We pick up the maximum score from one of these 4 and fill this matrix. And for the right plot, for the x-axis is like the index of each column, and the y-axis is the maximum score from each column. If we find like the maximum score of a specific column is like significantly smaller than a maximum score we ever reached, by significant score smaller meaning like larger than a preset threshold, then we stop the excitation and generate alignment from the maximum score to the like where it started. So if, for example, if we have some like homologous region have a like very high sequence diversity, maybe this kind of region have this kind of score drop very quickly, or maybe we have some like a very long indels. For example, the transposable elements, we also have this kind of max score drop quickly. So this kind of approach could not align across the kind of long indels or highly diverse homologous sequence. And also another problem we find is like in general, we use this kind of effing gap penalties to perform alignment. So if you ever look at into the parameter, for example, BLAS or BWA, they all have those kind of parameters. And if we check the like the indel length distribution between maize, between human and chimpanzees, we could find that there are like some indels have like a long indel also have a high frequency. These kind of long indels are generally result from the present or absent of transposable elements. And for the short indel, they are from a lot of different kind of mutations. So the long indels and short indels are result from different mutation mechanisms. If we optimize the gap score for the short indels, then we could not align the long indels very well. If we optimize the score for the long indels, then we couldn't align the short indels very well. So it's not possible to optimize the gap penalty to align short indels and the long indels at the same time. If we look at into the plants, of course, I mean, as Michelle introduced before, the plants have like much higher transposable elements activity and also have much longer indels. So we don't think that this kind of approach could align the long indels and short indels well at the same time. To solve this kind of problem, we developed a software called AnchorWave. The AnchorWave takes the reference genome, query genome, and also the reference genome annotation in GFF format as input. Firstly, we perform alignment for the protein coding sequence, and then we develop longest path algorithm to find the collinear regions. We develop 3 versions of longest path algorithm. So first one, we really perform end-to-end like collinear identification. We assume there's no inversion, no translocation. That's for each pair of homologous chromosomes. And for second one, for each pair of homologous chromosomes, we assume there may be like inversion, but we assume there's no translocation. And for the third one, we assume there may be like the translocations, inversion, and even fusion relocations, or maybe there's no like a homolog chromosome anymore. And then we use like a two-piece F gap cost strategy and also global alignment strategy to perform base pair resolution alignment for each anchor and inter-anchor region. And then we concatenate the base pair alignment in each collinear block together and output alignment in the map format. However, we find some like the inter-anchor regions are super long, and if we perform the base pair alignment, it costs a lot of CPU time and also a lot of memory. To reduce the cost, we try to find some novel anchors in the long inter-anchor region. I try to break those into shorter fragments to reduce the CPU time cost and the memory cost. Here, I also would like to introduce a little bit more about the two-piece F gap cost strategy. So for this strategy, we introduce another gap penalty parameters comparing to the like widely used this F gap cost strategy. And we optimize the first piece of F gap cost penalty parameter for short indels, and for the second piece of parameter for long indels. If you look at into this score, this is plot, the x-axis is like the indel length, and the y-axis is like a penalty, and we always use a smaller penalty as like a penalty for the final sequence alignment. And for the like the red line is from the first piece of gap penalty parameters. We could find like for this small like shorter indels, this is kind of first piece give a smaller penalty, and for the longer indels, the second piece of parameters give a smaller penalty. So we test our like software. I mean, actually, we did not use the real simulation data because we think the simulation data could not really recover the complexity of the real sequence evolution. Instead, we use like a variant coding of 18 Arabidopsis Synteny accessions. So here is how the variant coding looks like. So for the first column is a chromosome, the second column is like a position, and the fourth column is like a reference value, and the fifth column is an alternative value. So we replace the reference value with the alternative value, so we could generate the benchmark sequence alignment and also synthetic genomes. If we align the synthetic genome with the reference genome and compare the generated alignment with the benchmark genome alignment, so we could evaluate the performance of the genome alignment softwares. So here we perform alignment using our AnchorWave and also the Minimap tool, and also use LAST, also Marmer, and also GS align. We calculate recall, which is like the whole what's the proportion of the benchmark alignment could be recorded by generated genome alignment. We could find that the AnchorWave give us the highest record, and also we calculate precision, which is like how many of the alignment in the generated alignment could be identical in the benchmark alignment. We could find the like a minimum to give us highest precision. However, the AnchorWave give a second highest. And we also calculate F-score, which a single measurement combine the required precision. AnchorWave will give us a highest F-score. So it depends on which parameter you would like to measure the performance of the genome alignment approach. The AnchorWave is the best one or second best one, which is like comparable with the really best one. Next, we'll hand out to Michelle.
Okay, so we were interested in testing AnchorWave on genomes other than Arabidopsis, especially because Arabidopsis genome is a rather small plant genome. and also doesn't have that many transposable elements. So we wanted to think about ways to test AnchorWave for the goal of identifying presence and absence of indels generated by transposable elements. And so we did this using the maize genome and using a simulated maize genome, where we removed about 60% of all of the LTR retrotransposons from the B73 genome to generate a simulated genome. And now notably, we didn't remove all of the LTR retrotransposons from the genome because we didn't want to make this too simple of a problem. We wanted to leave some of them in there so that there still is the ability for these LTR retrotransposons to find other LTR retrotransposons to align to, kind of at the wrong sort of location. And so what we were assessing was whether, so we generated this simulated genome, and then we aligned these two to one another. So in the top right, LTR retrotransposons are kind of these longer types of transposable elements, the majority of them being between about 6 kb to about 15 kb. And then we took the reference genome and this simulated genome, it was a TE deleted genome, and aligned them to one another using the same genome alignment software that were shown in the last slide for Arabidopsis. And so then we just simply asked how many of these TEs that we deleted are actually called as a deletion across that entire region where we removed it from. And for AnchorWave, we see that we have a very high proportion of all of these TEs removed are able to be identified as deletions. Minimap2 recovers a little more than 20% of the TEs we deleted. And most of the other genome alignment software doesn't really tell us anything, is really incapable of calling these indels. And we think that this is probably because Minimap2 is the only one of these software besides AnchorWave that implements this two-piece apping gap cost to allow and deal with some of these longer indels. In fact, Minimap2 is kind of where Baoxing got the inspiration for it from. Okay, so AnchorWave is doing great on those indels, but the problem being that the indels are, it's kind of a simple case. We don't have any SNP variation or shorter indel variation in those flanking sequences. So as long as like you can call that position, we don't really have any other harder things to do. And so to assess real data, I'm going to first introduce how we're categorizing nucleotide differences into these 3 different categories. And I'm going to show this here now on this slide, but we'll also kind of have this as a little cartoon down in the bottom corner on the slides it's relevant for. And so in order to categorize kind of different types of sequence diversity on a base pair level, I'm going to highlight the difference between the reference and the query. And so everything we're talking about is going to be relative to the reference. So we can assign sites to position match sites, these orange sites, when a nucleotide is aligned to another nucleotide. Now these can be sites where the nucleotide is identical between the reference and query, like these first 2 cytosines. It can also be a position where there's a mismatch between the reference and query. And so in addition to these position matches, in addition to position match sites, we can identify gap sites. So these are sites where a nucleotide in the reference is aligned to a gap in the query. I'll note that, for example, there's this gap in the reference and an insertion in the query. From the perspective of the reference, we're not cataloging this, but Baoxing has developed approaches that allow us to kind of flip what we're considering as the reference and what we're considering as the query without having to do all of the computationally intensive alignment steps again. So it's kind of, everything I'm talking about here is very reference, or is anchored, anchor is a wrong word for this talk, focused on the reference. Okay, so now we have position match sites and gap sites. Aligners can also report unaligned sites, which are shown here in blue, where a base pair in the reference just isn't, there isn't a reported alignment in the query. Okay, so this is how we're categorizing these sites. And so we wanted to then align two maize genomes to another. And then we're gonna start the focus over on the plot on the left. And these are the results from the alignment of the B73 genome to the MO17 genome. And so across these multiple aligners. And so the first thing I want to point out is the proportion of these that are position match sites for each of these aligners. All of them get about 60 to 70% of the B73 genome that can be aligned, where a nucleotide can be aligned in B73 can be aligned to a nucleotide in MO17. And so this is kind of in line with what we expect based on what we know about kind of the structural diversity between maize genotypes. The major difference here that we can see between AnchorWave and the other aligners is the proportion of the B73 genome that's not aligned as position match. In AnchorWave, these are aligned as gaps, whereas most of these other aligners are reporting these as unaligned sequence. And so I'm going to highlight why this can be important now, focusing over on the right by using an additional data set to give us information about this. And so this is using a previous publication from Sarah Anderson in 2019, looking at presence-absence variation of TEs between different maize inbred genotypes, including B73 and MO17. So what this gives us then is which transposable elements that are present in B73 and are absent in MO17. And so I'm going to walk us through kind of what we would expect to see if this truly were the case. So we're going to kind of walk up these three possible lines here above this. So this first one is what AnchorWave is reporting, where we have position match of this flanking sequence, and then a gap, and then a series of gap characters across the length of the retrotransposon. And so that's what's reflected there. What many of the other aligners are reporting is kind of shown on the next line up, again with the flanking sequence matching, but this LTR retrotransposon sequence not being reported as aligned to anything. And so at first glance, this might not look all that different from just either reporting it unaligned or reporting it as a gap. But what is useful and lacking, so what's lacking in these unaligned sequences is we don't have any indication that kind of this left side of the flanking sequence necessarily matches at the same position as the right side of the flanking sequence. And so we lose some information here. Additionally, I want to highlight why I think that some of the differences between aligners in the position match sequences, these nucleotides that are getting aligned to one another, are probably wrong for many of the aligners other than AnchorWave. And so what's happening in some of these are this crosshatched region where TE sequence is being aligned to other TE sequence. And so presumably this is wrong. We have previous evidence that this TE just isn't present at this position in MO17, but when this gets aligned somewhere else, this is giving us kind of a false answer. So this crosshatched part is where there's polymorphic TEs that we don't expect to be present, but are getting aligned as nucleotides. And so the extent of this orange crosshatch portion in non-AnchorWave aligners is leading us to believe that AnchorWave is giving us a better alignment because it's minimizing those false alignments. Okay, so these are two maize genomes. Oh yeah, I wanted to highlight why we think AnchorWave is doing a good job of the regions it does align. So we're getting near complete genome alignment of the entirety of the genome can be found in these collinear blocks between MO17 and B73, as well as if we just take of these position match sites, these 60% of the genome that can be where a nucleotide can be aligned to a nucleotide, we can see that that's about 96% sequence identity. And so this is pretty close to what we've seen kind of historically in comparisons between maize genomes, especially those that have been focused on genic sequence. So like kind of often say something like 98.5% of the genome is the sequence identity between two maize genotypes. This is a little bit lower than that, but we're including a lot more sequence than just genic and near genic regions. So we're pretty excited about what this is telling us. Another comparison of maize genomes that can be made is, we know that there's a lot of structural diversity within the maize genome. AnchorWave can deal with things like inversions. So here we're looking at a comparison of the longest path between the B783 AB10 genome assembly on the x-axis and the B783 genome assembly on the y-axis. Now, this B783 AB10 is a inbred line that's been generated by recurrently backcrossing individual with abnormal chromosome 10, which carries the meiotic drive locus into a B783 background. So we expect, except for that focal locus on chromosome 10, there to be near perfect alignment across all of these chromosomes, which we indeed see. Additionally, we recover the two nearby inversions on chromosome 10, where the kinder locus is located. And I'll note that for right now, we aren't generating we're just able to get gene level resolution of these inversion breakpoints. Baoxing has worked a little bit on generating a base pair alignment across those breakpoints, but it's pretty difficult computationally, especially due to the fact that inversions tend to form in pretty structurally complicated regions of the genome. So it's where it's at for now. Okay, so everything we've talked about so far has been comparing two genomes or multiple genomes within a single species. But AnchorWave is a lot more flexible than just looking within species. We can look at, and some of the power is coming from being able to compare relatively divergent species, especially those that have had different levels of whole genome duplication. So like Baoxing I mentioned before, the sorghum genome and maize genome differ by a whole genome duplication, where after divergence of the sorghum lineage from the maize lineage has kind of formed by this allopolyploidy event, bringing together two semi-divergent genomes into one. And so what that means is that we can tell AnchorWave that we know this to be a fact, and then allow any anchor in the sorghum genome along the x-axis to be present up to 2 times in the maize genome. And so we can see that working in this longest path on chromosome 1 of sorghum over on the left, where there's a homologous chromosome or a homologous sequence on chromosome 1 of maize, but also chromosome 5 of maize and chromosome 9 of maize. And so AnchorWave is allowing us to kind of use that previously known information. We can also kind of, if we don't know about whole genome duplications that differ between our species, we can make this plot like this and make some of these inferences based on kind of the gene anchors that you observe. And so there's some nice documentation in the AnchorWave walkthrough about how to do that. Okay, so this is the anchor part, but now we want to do the wave part, the actual base pair alignment of maize and sorghum genomes. And so here I'm going to present kind of 2 biologically informed prior expectations of how a sequence aligner should work. One of the problems that we kind of kept running into was the fact that if you give an aligner 2 sequences, it's going to give you an output. And it can be really tricky when you're using real data and not simulated data to assess whether it's doing a good job. Like it's easy enough to kind of look at a single locus and say, okay, this looks right to me or this doesn't look right, but scaling that up genome-wide can be pretty tricky. So I'm going to talk first about a case we think is a positive, where we where we have a sequence that we expect to be conserved between these two species and ask whether it truly is. And then second, I'll talk about a case where we don't expect sequences to be conserved and they aren't. Okay, so this first case of sequences that we expect to be conserved between maize and sorghum is going to focus on transcription factor binding sites. Now, this maybe is a kind of secondary, so maybe the first thing that we would think about for conservation between species would be genes. But because we've used these gene anchors to define where alignment is initiated for AnchorWave, we think that's kind of cheating to assess whether it's doing a good job of aligning those sequences. So what I'm showing on the left is a plot much like we saw for maize, where we have our aligners along the x-axis and the proportion of the genome as a position match, these nucleotide to nucleotide alignments. AnchorWave recovers about 8% of the maize genome in these position match sites with sorghum. Some of these other aligners, like last, are identifying up to a quarter of the maize genome being aligned to sorghum. And again, a major difference between AnchorWave and these other aligners is that AnchorWave is producing these gapped regions of alignment. Okay, so 8% versus 25% is a pretty big difference in the amount of the genome being aligned. And so we wanted to assess whether last was just kind of over-aligning sequences to one another. And the way that we came up with doing this was transcription factor binding site alignment. So here we're using transcription factor binding sites from over 100 ChIP-seq data sets as they're measured in the maize genome. And we're going to start with the x-axis here, just looking at transcription factor binding site recall. So here we're just asking how many of those transcription factor binding sites in maize are aligned to matching nucleotides in sorghum. And so we don't have like a perfect biological expectation of how high this value should get. We expect some conservation of transcription factor binding sites, largely because there are genes between maize and sorghum that are conserved and expressed in the same developmental stage, cellular compartment, all of these sorts of things. So we definitely don't expect this number to be down at 0, like is recovered for many of these mini-map aligners with the triangle. We also probably don't expect it to be up at 1. We don't expect every single maize, all of maize regulation to be exactly the same in sorghum. But what we do see is that AnchorWave is near the top here. You might not actually see it because it's up this circle up here on the higher up on the y-axis. But we're recovering over half of the maize transcription factors binding sites in sorghum. Okay, so that's all in good, but AnchorWave isn't actually doing all that much better than last in this comparison. And so we also wanted to kind of look at a metric comparing whether, so it would be really easy to align all these transcription factor binding sites if you just reported false alignments for all of them. And so we wanted to ask whether there was an enrichment for transcription factor binding site alignment over that expected in the genomic background of non-transcription factor binding site sequences. And so that's what this y-axis is summarizing. So for most of the other genome aligners, there's no enrichment for these transcription factor binding site alignments. They're right about 1. Whereas AnchorWave is getting us over 10 times the alignment rate in these transcription factor binding sites than it is in other regions of the genome, which indicates to us that we're kind of in this optimal zone of identifying real conserved sequence in this 8%. Okay. So that was kind of the positive. We think that AnchorWave is not over-aligning sequences. We also wanted to look at sequences that we don't expect to be aligned between maize and sorghum. And in this species comparison, that's the transposable elements. And that's coming from the fact that transposable elements have been active along the maize lineage, And we can look at the age of the timing of insertion of those elements. And the vast majority of them have inserted in the last couple million years, much more recent than maize and sorghum have split 12 million years ago. And so this means that if we observe a transposable element insertion on the maize lineage, there's no real way for that to be shared at that same position in the sorghum lineage. And so that sequence should not be aligned to a base pair in sorghum. And so indeed, when we look over on the right here again, we think, so the cross-hatching again is TEs in the maize genome. And so for AnchorWave, we see that many of these are in the gap and unaligned sequence. Whereas for this last alignment, a large proportion of the position match sites are actually in TEs. And so we think that these are false alignments, where last is kind of aligning a TE to a TE somewhere else in the genome that it really shouldn't be aligned to. And so again, this is kind of our negative control telling us that AnchorWave is kind of right in that spot that that we trust for, based on these biological expectations for thinking about alignments between maize and sorghum. Okay, so everything we've talked about so far is really maize focused, which maybe for Z evolution is fine, but we also want to highlight that, highlight that that AnchorWave works on things other than just maize. So here I'm going to show a summary of alignments that we've compared of starting with maize, and kind of help to allow you to maybe guide to think about whether AnchorWave is the appropriate software to be using to compare genomes that you are interested in. So I have a phylogeny of these various maize relatives over here on the left, with red stars showing where there's been a whole genome duplication event. And in the middle in purple is the proportion of the genome that's in collinear blocks. So I've already shown you this comparison between maize and maize. So B73 on the top is going to be compared to each of these genomes below it. So this other maize individual is Mo17, where we recover almost 100% of the genome in collinear blocks. As we kind of move out sorghum, there's less of the genome in these collinear blocks. Maize versus rice is just above 80%, whereas maize versus banana gets us down to only 11% of the maize genome can be found in collinear blocks with banana. Largely, this is due to the fact that there have been 6 rounds of whole genome duplication that separate maize from banana. And so this is breaking up some of this ability to call collinear blocks. And so this might be a case where AnchorWave is kind of a limitation of AnchorWave and its ability to align these genomes. Another thing that I do want to highlight is you can look at this nucleotide identity at these position match sites. So this is essentially sequence identity of aligned sites. And so we can see that this kind of goes down with evolutionary time, as we might expect, with the 96% I highlighted for B73 versus Mo17, and kind of all the way down to about 60% for maize versus banana. We can also look at, so this has all been kind of still maize focused. We can also look at dicot comparisons of Arabidopsis versus some of these other species that differ in whole genome duplications. And again, see a contribution of both kind of evolutionary time and the number of whole genome duplications, reducing some of this, the proportion of the genome that we're really able to align. So because AnchorWave is relying on collinear blocks, this is an important thing to keep in mind when you're deciding on a genome comparison aligner. We can also look at some different polyploid taxa. Sorry, it's not coming. There it is. We can also look at other polyploid taxa and see that it's not always just evolutionary distance, and that there's a lot of complicated things that go on in genomes. So for example, this tomato versus potato comparison about 70% of the genome can be found in collinear blocks with a 7 million year divergence. However, soybean versus common bean has a 20 million year divergence, but more of the genome is retained in collinear blocks. And so kind of keeping these things in mind and kind of looking at some of these differences is important for thinking about whether AnchorWave is the appropriate genome aligner for species. But we think in a lot of these cases that this is really doing a lot better than many of the prevalent common things people are using these days. So now we have our takeaway once my slide comes up. Yeah, and so kind of if you're aligning a genome with a whole genome duplication or a lot of TEs or dispersed repeats, I encourage you to give AnchorWave a try. Baoxing has a lot of really nice documentation up that'll walk you through everything from downloading the genomes to actually producing the base pair alignment and interpreting it, available here. And we would like to thank our funding sources. Sorry, this slide is slow for me, so I hope that I'm coming across. So we'd like to thank our funding sources from USDA and NSF, as well as co-authors from the Buckler Lab, Ed and Lynn Johnson, a programmer, and the WFA or wavefront algorithm development team helped us to implement the 2P's gap mapping cost into that, Santiago and Michael. And with that, we can take any questions.
All right, thank you very much, you too. We have some questions in the chat. I'll start with Nathan. Nathan, you want to just unmute and ask your question yourself?
Sure. I think the answer is probably no, this doesn't work well, but as you're talking about things, I'm curious if you can use long reads or exceptionally long reads and use AnchorWave to call indels, or essentially another way of asking this question is how small of a segment do you need to have enough anchors before you can use this for calling indels?
So for the default parameter, we align the reference gene to the query gene. If I mean by default, if there are 3 collinear genes, we could identify that as collinear blocks. So we defined this kind of collinear block by using the number of genes. We didn't really care about how many base pairs is that. So it depends on what kind of genome you are working on, how dense is the gene in your sequence. So, yep, that's it.
Yeah, I think also sorry. I think also sorry. If you have those ends and you know where they are, you could use the wavefront algorithm and the 2P's gap affine stuff that we've developed to align that internal part in a global alignment strategy. And I don't know, I have liked the output of that so far, so it might be possible to modify.
Can I ask a follow-up on this? So how dependent is it then on the quality of the assembly of those 2 genomes? And you say genes, so is it dependent on the annotation of what you call the reference, or?
Yes. I mean, it depends on the input files are the different genome reference annotation, query and a query genome. So if you don't have very high quality reference genome, a reference genome annotation, that doesn't really matter. I mean, we only use this kind of annotated gene as an anchor. If you could annotate some protein coding gene as some conserved sequence, then that's okay.
Okay, thank you. Then we had some questions by Jeff. You want to unmute and ask?
Yeah, thanks. First, fantastic presentation. I liked the comparison of plant and animal genomes, the Synteny and the comparison of different plant genomes at the end. The 4% where you compared two maize genomes and said they're identical sequence 96%, is that 4% difference including indels, or is that just base pair substitutions?
Including indels.
Including indels, okay.
Well, I think that number is just base pairs. So that's position match sites. And so that's the number of nucleotides that are mismatched divided by just the total number of nucleotides that are aligned to nucleotides.
But does a mismatched nucleotide include a single base pair indel?
No, but I don't know. That was my interpretation of it, but if Baoxing's saying the other thing, then I'm probably wrong.
I'm not sure you're talking about the same thing. You're talking about this 4% or?
No. Michelle had a slide which showed two maize genomes and said they're 96% identical at the sequence level.
Oh, okay.
Yeah, so 96.05, that means 4% of the sequence is different. And I'm wondering if that includes, like on the bottom right there, the C versus indel. So is that does the mismatch include single base pair indels or short indels, or is it only different nucleotides?
This is just different nucleotides from the perspective of the reference genome. So any of these orange sites, this is just the number of mismatches of orange sites divided by the total number of orange sites.
So your estimate of average pi in the maize genome is 4%.
Yes.
Cool.
You had more questions, Jeff? You just want to keep going? Or?
Oh, yeah, sorry, I forgot. I'm still trying to get my head around 4% sequence divergence. So in the phylogeny, you compared to banana and cited limitations of AnchorWave, but like how well does last Z or Minimap do in those sorts of approaches, in those sorts of comparisons?
Not great. I'm trying to remember the details. We didn't do all of these comparisons for every taxa. A lot of this, like the computational time required to run last Z on some of these more complex comparisons, like we couldn't find computers that could do it in reasonable amounts of time. I mean, yeah, I think like maize versus banana, if you're really committed to aligning maize to banana, that like the portion of the genome that AnchorWave can align probably is going to give you results that like are interesting. But I mean, I think that there probably are like orthologous genes between maize and banana that if you're really just caring about gene evolution, you might be able to recover that more appropriately for different ways of from just grabbing the genes rather than relying on these at least 3 genes in a continuous collinear block.
Thanks.
Okay, Ed, did you had some comments on Jeff? Do you want to add on this? Or? Okay, and does anybody else have a question? Any other questions? That seems to be not the case. Then thank you two very much again. And thank you all for joining.